In this video, we will discuss fluorescence, its observational techniques, fluorophores, and the properties of these fluorophores. What is fluorescence? Fluorescence is the process of light emission by a substance when its electrons return to ground state after being excited by light or an electromagnetic radiation source. This process is widely used in organic chemistry, as well as biological chemistry, for its ability to help track movement of organelles, molecules, and also providing information about their structures, environment, and reaction properties. Now we will discuss some observational techniques, such as the stimulated emission depletion microscopy, photoactivated localization microscopy, and how these discoveries make an impact on our views on fluorescence. Stimulated emission depletion microscopy was discovered by Stephen Hell. The stimulated emission depletion microscopy surpassed the limit of light microscope and created a better resolution. This microscope uses two laser beams, one that stimulates the fluorescent molecules and creates a small lateral offset on the excitation beam. This method increases the resolution by a factor of 4.5 by using a stimulated emission. The emission is based off the excitation energy of a typical fluorophore. In this diagram, you can see the setup of the microscopy with two beams canceling out the fluorescence to get a better resolution. This diagram depicts the excitation levels of a typical fluorophore that is used for the emission beams in the microscope. The energy levels that are involved in the excitation of a typical fluorophore can be seen here. Photoactivated localization microscopy, also known as PALM, was discovered by Eric Betzig. The optical fluorescence transition microscopy helps get a closer look at a molecular scale. The photoactivated localization microscopy can obtain precise localized molecules. It uses thin sections instead of bulk samples, so it will exhibit less autofluorescence, therefore getting a better resolution. Also, it magnifies the small sections so that one could see more depth on a molecular level. How do these discoveries make an impact on fluorescence? The diagram shows the difference of a conventional fluorescence microscope compared to the 3D picture. As you can see, the picture has a clearer resolution. By improving the light microscope, it can further science into finding cures for diseases. Also, the use of quantum dots will help improve the resolution of a 3D image in cells by allowing brighter emissions than regular dyes. A fluorophore is a fluorescent chemical compound that emits light when excited. Fluorophores are sometimes used alone as dyes for staining structures, as a substrate of enzymes, or as a probe or inhibitor. Most of the time, it is bonded to a macromolecule as a marker for bioactive reagents. These fluorophores are used in fluorescent imaging and spectroscopy. There are two types of fluorophores, intrinsic and extrinsic fluorophores. Intrinsic fluorophores are fluorophores that are already bound to a molecule. These fluorophores consist of aromatic amino acids. The most popular aromatic amino acids are tryptophan, tyrosine, and phenylalanine. These amino acids are the main proteins responsible for absorption and fluorescence of proteins in the UV. Out of the three, tryptophan is the most sensitive to the local environment. In solutions, however, tyrosine is more fluorescent. This is because in a protein, the tertiary structure inhibits the fluorescence of tyrosine as well as an energy transfer from tyrosine to tryptophan residues. Extrinsic fluorophores are manually attached to a molecule. Extrinsic fluorophores include fluorescin and rhodamine, naphthalene sulfonates, nucleic bases, ion detectors, carbohydrate fluorescent probes, oxidation of tryptophan residues with N-bromosuccinimide, and nitration of tyrosine residues with tetranitromethane. Fluorescein and rhodamine covalently bond to cysteines and lysines of proteins. Naphthalene sulfonates bind covalently to proteins and are used to follow their conformational changes. Nucleic bases, such as puric and pyrimidic and guanidine bases, are fluorescent, guanidine being the strongest of the three. Their fluorescent lifetime is weak, and they are polar as well. Ion detectors are fluorophores that detect the presence of a substance. For example, calcium cells are detected when Fura2 or Quin2, which are types of fluorophores, bind to calcium or other proteins in cells. Carbohydrate fluorescent probes are fluorophores that covalently bound to small carbohydrates and usually a protein in order to see the interaction between the carbohydrate-protein interaction. The oxidation of NBS to proteins allows the titration of tryptophan residues and helps clarify the energy transfer within the protein. An important factor to notice about NBS is that it may change the conformation and structure of protein, therefore affecting the fluorescence. The nitration of tyrosine residues with TNM has effects towards phosphorylation and dephosphorylation of tyrosine. The chemical nitration of tyrosine residues on proteins was often found to inactivate and alter the properties of specific enzymes, and is used to observe the subsequent effects on cell proliferation. Properties of fluorophores are lifetime, emission spectra, and intensity. The lifetime of a fluorophore decays exponentially at about 1/e of the original amount of excited molecules and absorbs energy between UV to visible light. This process emits fluorescence that has energy range between visible light and infrared in a matter of nanoseconds through emission, which is shown in a Jablonski diagram. The intensity of fluorescence depends on the quantum radiation yield of the fluorophore. In other words, how much an excited electron drops in energy level when it is relaxed. A simplified Jablonski diagram shows electron transition between ground state to excited state. In the excitation process, light excites ground state electrons to a higher energy level. Then, at the emission process, the electron relaxes back to lower energy levels, producing fluorescence emission. How far of a drop in energy levels will depend on the quantum yield of the fluorophore radiation. This difference creates different wavelengths and color intensities. Looking at the big picture, the difference in wavelength between max absorbance and emission is caused by Stokes shift due to the loss of vibrational energy in the excited state. Other properties of fluorescent molecules are pH and viscosity. Some applications of fluorophores require pH sensitivity, such as making dyes with different solvent. Dipole properties allows attraction and association of molecules of a solvent with molecules or ions of a solute. Viscosity, fluid viscosity in this case, can be chemically modified to fit its environment, thus having an electron donor and an electron acceptor component. Fluorophores by themselves are not perfect because errors such as noise from other molecules in the environment, low fluorescence emission, and imaging can be obtained. Therefore, some ways to improve fluorophore properties are the use of dyes, more dipole-to-dipole interactions through pH modifications, improving fluorescence emission by reducing dye concentration noise, and refractive index. Lastly, fluorescent molecular photoswitches can be used to turn on or off a fluorophore and allows direct measurement of the target molecule with reduced concentration noise. This process is called photoisomerization. Application of fluorophores. Since we know that the properties of fluorophores are, here are some useful scientific applications. Fluorophores can be used as a probe salvation for protein labeling. They can also be used to analyze cytoplasmic fluid in the examination of diseases that cause changes in cell signaling. Fluorophores also allow imaging at a nanoscale level possible. and they can be used to gather more light energy.